welcome back, insignis. Buenas tardes, amigos, amigas. Magandang hapon sa inyong lahat. Shana Tova to all my friends who's celebrating Rosh Hashanah. So for today, we're going to talk about part 2 of our exam 2 review, specifically biochemistry, right? So first, we need to know what happens to glucose. So glucose is very important, right? It gives us energy. It gives us ATP. So glucose can be metabolized into different pathways. Anaerobic metabolism to convert it to lactate, TCA cycle to convert it to H2O and CO2, HMP shunt to convert it to ribose and NADPH. We can also do fatty acid synthesis to create fatty acids and glycogenesis to make glycogen. You can see glucose is a very versatile molecule. So how does the glucose enter the cell? So GLUT1, insulin independent, meaning this only happens when glucose is very high, okay? So GLUT1 can be seen in brain and your RBCs. GLUT4 is insulin dependent. This is commonly expressed in your fat tissue or your skeletal muscle. GLUT2 is insulin independent. This is bidirectional. Most of the tissues in the body has GLUT2, right? Liver, you have the kidney, you have the intestine, and your pancreas. So again, GLUT1, they only get glucose when you have a high concentration of glucose in the blood. So your brain and your RBCs, they have GLUT1 receptors, insulin independent. GLUT4, they are insulin dependent, right? So the uptake when the glucose is low. The receptors can be seen in fat tissue and skeletal muscle. So when you're doing exercise, right, you are increasing the GLUT4 receptors on your skeletal muscles, thereby increasing glucose uptake. So sometimes you feel tired after doing exercise. That's the reason why. You can have transient hypoglycemia. GLUT2 is expressed where? Liver, kidney, intestine, and your pancreas. It is insulin independent. It doesn't, meaning they only uptake glucose when the concentration is high. This is a bidirectional receptor. It can be used in gluconeogenesis. And remember, in the intestine, the glucose is out of the epithelial cells to your portal vein. So this is glycolysis. So we're breaking down glucose and making pyruvate. Do I need to know all of those intermediary steps? No. The only important thing here is glucose, I think, fructose 1,6, bisphosphate, and pyruvate, the end product of your glycolysis. So first, we need to convert glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate. We are now in glycolysis. Now, high yield, you need to know the difference between hexokinase and glucokinase. This is the enzyme that catalyzes the conversion of your glucose to glucose 6-phosphate. So when you have low blood sugar, hexokinase is working. Okay, so you have glucose to your tissues. You want the glucose to go to your tissues if you have low blood sugar and not to your liver. However, when you have high blood sugar, the glucokinase is working, right? So that the liver will store your glucose as glycogen. So question, in terms of Km and Vmax, how do we describe hexokinase versus glucokinase? Anyone?
Hexokinase has a low Km and low Vmax, and glucokinase has a high Km and a high Vmax.
Excellent. So think about it this way, right? So low blood sugar, you're not eating, you're fasting. What is constitutively active? Your hexokinase is working. Okay? So the glucokinase is not working. This is inhibited by glucose 6 P, right? In low blood sugar, the glucose goes to your tissues. You don't want it to go to your liver. If you eat a lot and you have, after a meal, you have a high blood sugar, glucokinase is working. You have high glucose. Remember, it is high insulin. In the presence of insulin, glucokinase is working. So you can say that it is insulin dependent. Why do I need glucokinase to work? Because I need that sugar to go to the liver to store it as glycogen so that I can have energy in terms of fasting. Okay? So that's your glucokinase and your hexokinase. Any questions so far? Or you're good to go? All right.
I think there was a question in the chat. It said, someone said, doesn't glucokinase kind of depend on hexokinase to jumpstart it?
They're different though. So it all depends on whether you have a low blood sugar or high blood sugar. Or in other words, it all depends when insulin is active or insulin is not active. Okay? Most of the time, hexokinase is working. But in the presence of insulin, for example, a high blood sugar, glucokinase is working. So it depends upon that system, if it's low blood sugar or high blood sugar. That's the first condition, whether it's fasting or fasted state. If it's low blood sugar, you're in the fasted state. So which one is active? Hexokinase is working, right? Why do you think this is hexokinase is working in low blood sugar? Because you want glucose to go to the tissues. You don't want to store the sugar. When you are in the, after you finish a meal, you have a high blood sugar, most of the time glucokinase is working, right? Why? You have high glucose and insulin. When you think about insulin, think about anabolic reactions. You want to build something. Where does this blood sugar needs to go? I have too much. Well, I need to build something. I need to store it to the liver as glycogen so that in the future, if I am in a fasting state, I can break down that glycogen. Okay, so hexokinase can be seen in skeletal muscle, right? Glucokinase can be seen in the liver, the pancreas, okay? Hexokinase, as someone have said it, lower Km, lower Vmax. Therefore, at fasting, glucose levels, remember, low blood sugar, hexokinase is at Vmax. Glucokinase has high Km and high Vmax. High Km meaning it spares glucose stores for hexokinase in the brain, muscle, and other tissues. Glucokinase is also a glucose sensor. Okay? At fasting glucose levels, the activity varies based on the blood glucose concentration. And the regulation is allosteric. Hexokinase, it is inhibited by glucose 6-phosphate. So the substrate here, the product. If this is too much, hexokinase is inhibited. Glucokinase, on the other hand, is activated by insulin and inhibited by glucagon. I think you're going to learn, I don't know if they talk about this one, that if the glucokinase is bound to a regulatory protein, it is inactive. So you have the glucokinase, what is that? Glucokinase regulatory protein, right? If it is bound to that one, it is inactive. So those are the things that I want you to know about the hexokinase and glucokinase. Know the tissue distribution. Know the kinetics, meaning the Km and Vmax, what happens to each, and know the regulation. Allosteric, which is hexokinase, and hormonal, which is your glucokinase. Those are the most important things that I wanted to know between the two. Okay? We're clear so far? Is that explanation helpful or did it make it worse?
It was helpful. Thank you.
Okay. So please note those. Those are high yield points. So what if I don't have glucokinase? Is this bad? Yes, remember, it's a sensor. It senses your blood glucose concentration. So if I have a glucokinase deficiency, what happens? I cannot sense increase in blood sugar, right? It results in what? Hyperglycemia. So your pancreas, remember glucokinase is expressed in your pancreas. And in your liver also. So the pancreas gets less sensitive, so you can have mild hyperglycemia. Remember, if you have a glycokinase deficiency and you're pregnant, it can exacerbate this hyperglycemia. Okay? That's your glucokinase deficiency. So how am I going to regulate glycolysis? Via PFK1, high yield. So here, you are converting fructose 6-phosphate to fructose 1,6 bisphosphate. What's the enzyme? PFK1. What are the inhibitors and inducers? When you think about inhibitors, think about high energy molecules. If you have increase in citrate and ATP, you don't need glycolysis because you have high energy. Citrate comes from your TCA cycle. You're creating more ATP, right? So you don't need energy. You don't need glycolysis. So these are your inhibitors. When you think about inducers, think about low energy molecules. Think about AMP. Think about fructose 2,6 bisphosphate. If you have low energy, meaning you have low ATP, you need to make more ATP. How am I going to make more ATP? You need more glycolysis. These are your inducers. Okay? So how do we regulate glycolysis? What's the rate limiting step? Phosphofructokinase 1. What inhibits it? High energy molecules. Citrate from the TCA cycle and ATP. What induces it? More glycolysis, low energy molecules, AMP and fructose 2,6 bisphosphate. Now, look here. Fructose 2,6 bisphosphate, in the fed state, you have increase in insulin. If I have an increased insulin, I have an increase in fructose 2,6 bisphosphate, which increases my PFK2, which increases my fructose 1,6 bisphosphate. Okay, so for fed state, you have increase in insulin. If you're eating, you're good to go, right? You need to store that. So you have an increase in fructose 2,6 bisphosphate. Okay, contrast this one with fasting state. If you have a decrease in insulin, you have a decrease in fructose 2,6 bisphosphate. You have a decrease in your PFK2. So you have a decreased conversion of your fructose 6-phosphate to fructose 1,6 bisphosphate. So what's the point here? The point here is whether you have insulin or not, meaning whether you are in the fasting state or not. If insulin is activated, you are increasing fructose 2,6 bisphosphate, which increases PFK2. So you increase the conversion of your fructose 6-phosphate to fructose 1,6 bisphosphate. If you have a fasting state, you are decreasing insulin. There is no insulin because you're fasting. Glucagon is reigning. So you have a decrease fructose 2,6 bisphosphate, decrease PFK2. So decrease conversion of your fructose 6-phosphate to fructose 1,6 bisphosphate. When insulin is increased, think about anabolic reactions. You want to build something, right? When glucagon is ruling, think about catabolic reactions. You want it to destroy something. Now, Another thing with glycolysis is your energy stage, right? So your pyruvate kinase is the end point of glycolysis from phosphoenol pyruvate to pyruvate. This is not reversible, right? So this is inhibited by your high energy molecules, your ATP and your alanine. This is also activated by a feed-forward activation of your fructose 1,6 bisphosphate. Where does this come from? It comes from here. Fructose 6-phosphate to fructose 1,6 bisphosphate via your phosphofructokinase. So an increase in fructose 1,6 bisphosphate, right? An increase in your PFK activity can actually increase your pyruvate, okay, via feed-forward activation. So in terms of glucagon and epinephrine, right? So it phosphorylates a kinase. So if you phosphorylate a kinase, it doesn't work anymore, meaning the pyruvate kinase is inactivated, which slows down your glycolysis. You don't need glycolysis in starvation, right? Glucagon or in a workout, your epinephrine is increased because you want to make glucose. So it slows glycolysis and it favors gluconeogenesis. Anything so far with glycolysis? So how many ATPs am I going to invest in glycolysis?
2.
2.
2, okay. How many the net ATP am I going to get in glycolysis?
2.
2. Okay. So we're good so far for this. So let's summarize again. Listen carefully. Okay. So what's the function of glycolysis? 1, generation of ATP from glucose via what? Substrate level phosphorylation, as opposed to your electron transport chain, right? To oxidative phosphorylation. Is this used by all cells? Well, most of the cells, yes, with oxygen, right? Remember, glycolysis is the only source of energy for your red blood cells, right? Because they don't have mitochondria. So how can they undergo citric acid cycle in your red blood cells? Glycolysis is in the cytoplasm, right? This is irreversible. What's the net reaction? Glucose plus 2 ADP plus 2 NAD plus, it yields 2 pyruvate molecules. As you've said, 2 ATP, 2 NADH. You have 2 hydrogen ions there and some water. And we talked about the enzymes. Don't forget, phosphofructokinase 1, this is the rate limiting step inhibited by ATP and citrate. It is stimulated by AMP and fructose 2,6 bisphosphate. Fructose 2,6 bisphosphate is synthesized by your phosphofructokinase 2. Don't forget that when you're in a fed state, increase in insulin, you have a decrease in cyclic AMP, you have a decrease in protein kinase A, therefore you have a decrease in fructose bisphosphate 2 and increase in PFK2, which increases your phosphofructokinase 1, which increases the conversion of your fructose 6-phosphate to fructose 1,6 bisphosphate. That's high yield. Let me repeat that again. Fed state, increase in insulin, which decreases cyclic AMP, which decreases protein kinase A, which increases PFK2, which increases PFK1, which increases conversion of your fructose 6-phosphate to fructose 1,6 bisphosphate. So what if I have in the fasting state? Then do the opposite of what I've said. If you're in the fasting state, you have an increase in glucagon, you have an increase in cyclic AMP, increase in protein kinase A. You have a decrease in PFK2. You have a decrease in PFK1. You have a decreased conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. No glycolysis. You have gluconeogenesis. What if I have a pyruvate kinase deficiency? This is most commonly autosomal recessive. What happens? No ATP, decreased ATP generation. What happens to my RBC? Poor RBCs, they swell and they lyse, right? What can we see in someone who has pyruvate kinase deficiency? Chronic hemolysis. However, contrast this one with G6PD deficiency. In G6PD deficiency, you have Heinz bodies there. You have bite cells. Here in pyruvate kinase, you don't have any Heinz bodies. All right, so that's just the gist of your glycolysis. Know the regulation, know the enzymes, know the inhibitors, know the inducers. Okay? Next one, Alanine cycle. Alanine cycle is when the skeletal muscles can degrade protein for energy. So we produce alanine. The alanine from the muscle, it goes to the blood. So you break down amino acids to alanine here from your muscle. It goes to the blood and then to the liver. And the liver is happily obliged to convert alanine to your glucose. Don't forget that in the liver, you have the urea cycle. So lactic acidosis. Lactic acidosis is if you have pyruvate and it doesn't go to the TCA cycle. You have the end product of your glycolysis, you have the pyruvate. Now, if you have decrease in oxygen, the pyruvate will go to lactate dehydrogenase. We're going to go to the lactic acid cycle, meaning you have an increased lactic acid production. You have acidosis, so decrease in pH. You have a decreased bicarb. So most commonly seen in alcoholics, actually. So it elevates your anion gap. Acidosis, don't forget from term 1, how to calculate your anion gap acidosis, right? Do you know the formula? Chloride minus sodium plus, okay. Revise this one then. This lactic acidosis, what can we see? We can see sepsis, you can see bowel ischemia, you can see seizures. End product of glycolysis is pyruvate. In the absence of oxygen, it cannot go to the TCA cycle. It cannot go to the mitochondria and undergo TCA cycle. Where does it go? Lactic acid cycle. What enzyme converts pyruvate to lactate? Lactate dehydrogenase. What can we see in a patient who has lactic acidosis? Elevated anion gap acidosis or metabolic acidosis. You can have sepsis, you can have bowel ischemia, and of course, the feared complication is seizures. Right? You don't want your patient to have seizures. We talked about pyruvate kinase deficiency, autosomal recessive, RBC is most affected. You can have extravascular hemolysis and splenomegaly. So let's summarize your glycolysis. So key steps, regulation. This is on the right-hand side, the complete pathway. They're going to test you on the regulations. Okay, so 1, hexokinase, glucokinase, right, from the glucose to glucose 6-phosphate. 2, PFK1, from fructose 6-phosphate to fructose 1,6 bisphosphate. And 3, pyruvate kinase, which converts phosphoenol pyruvate to pyruvate. Now, these 3, the regulatory steps, we also call this the irreversible steps. So meaning when you undergo gluconeogenesis, you have 3 different enzymes. Because these are irreversible, you need to have different enzymes for this one to undergo gluconeogenesis. We're going to talk about that later. Don't forget the regulations. They love the regulations. AMP, low energy. Fructose 2,6 bisphosphate, low energy. It increases your PFK1 to make fructose 1,6 bisphosphate. ATP and alanine, high energy. It inhibits your pyruvate kinase to convert phosphoenol pyruvate to pyruvate, thereby lessening your pyruvate. Again, ATP expanded. So someone said it, you invest to where? The first one, glucose 6-phosphate. This is either hexokinase or glucokinase. And then another ATP to spend here in your PFK1, phosphofructokinase 1. And then you generate 2 ATPs in the end. Here, 1,3-bisphosphoglycerate and 3-phosphoglycerate. And phosphoenol pyruvate and pyruvate. This is your pyruvate kinase here. Another ATP. So you invest 2, you get 2. What is the fate of pyruvate? We talked about pyruvate can be converted to lactate, right? Via lactate dehydrogenase. And then this lactate, it can go to the liver via the Cori cycle to be converted back to glucose. Or pyruvate can become acetyl-CoA and via pyruvate dehydrogenase. And acetyl-CoA will gonna go to your beautiful TCA cycle, which we're gonna talk about later, to make ATP. Pyruvate can also undergo gluconeogenesis via pyruvate carboxylase. If we want fresh glucose for our body, again, pyruvate can be converted to alanine via your aminotransferases. And alanine, as I've said, can be converted via the alanine cycle in the liver to make fresh glucose. High yield, know the fates of pyruvate. Uno can become lactate. Dos, acetyl-CoA. Tres, gluconeogenesis. Cuatro, alanine. Know the enzymes. Uno, lactate dehydrogenase. Dos, pyruvate dehydrogenase. Tres, pyruvate carboxylase. Cuatro, you have your amino transferases. Lactate can be converted in the liver to glucose via what cycle? Cori cycle. Alanine in the liver can be converted to glucose via what cycle? Alanine cycle. Acetyl-CoA can be converted to ATP via what cycle? TCA cycle. We're good so far with glycolysis and pyruvate metabolism?
Yes.
Okay. Next one. Gluconeogenesis. We need to make new glucose, right? This is now in the fasting state. So first, pyruvate needs to be converted back to phosphoenol pyruvate. So this is my trick. When you hear carboxylase, think about biotin, B7. When you think, yes, B6 is pyridoxine, right? So B6 is pyridoxine. Think about decarboxylase, think B6. When you think about carboxylase, think B7. When you think about dehydrogenases, think about B1, your thiamine. When you think about hydroxylase, anything that has hydroxylase in the name, think about BH4, your tetrahydrobiopterin. Okay, that's my cheat sheet to know the cofactors.
Could you repeat that one more time?
Okay, so if I have carboxylase, that's 7, B7, right? So you think about biotin for carboxylase. For decarboxylase, think B6, pyridoxine. When you think about dehydrogenase, think B1, thiamine. When you think about hydroxylase, think about BH4, tetrahydrobiopterin. Okay, those are just my cheat sheet. If I see that they end, there's a chemical that ends in that, I assume that the cofactor is always there. Okay, so the pyruvate, it needs to be converted to oxaloacetate via what enzyme? High yield pyruvate carboxylase. So you need biotin in there. From oxaloacetate, it can now get out of the mitochondria and go to the cytoplasm to be converted by PEP carboxykinase to phosphoenolpyruvate. So two enzymes here, that's very important. Pyruvate carboxylase via biotin, and you have your PEP carboxykinase, which needs GTP. Don't forget that on high yield, GTP for your PEP carboxykinase. So biotin, why is this important? See my keyword? My keyword for biotin is carboxylase. So this is a cofactor for carboxylation enzymes. So your pyruvate carboxylase, acetyl-CoA carboxylase, propionyl-CoA carboxylase. You can see they all end in carboxylase. So that's your biotin. It adds one carbon via CO2. So what if I have a biotin deficiency? If you're a trainer, you want to build muscles, right? So you want to consume egg whites, raw egg whites. So it has a component called avidin. This actually binds to your biotin, which decreases the biotin in your body. So high yield, massive consumption of raw egg whites, the avidin in the egg whites. Think about the bodybuilders, if you want to build muscles, right? So what can we see in a patient who has biotin deficiency? Dermatitis, glossitis, enlarged tongue, loss of appetite, and nausea. Okay. Next part. From fructose 1,6 bisphosphate to fructose 6-phosphate. What's the rate limiting step of gluconeogenesis? High yield fructose 1,6 bisphosphatase 1. We are converting fructose 1,6 bisphosphate to fructose 6-phosphate. Remember, we are trying to make glucose here. This is the reverse of your glycolysis. Again, PFK versus fructose 1,6 bisphosphatase 1. If you have AMP and fructose 2,6 bisphosphate, you are increasing the activity of your phosphofructokinase 1 in glycolysis. In gluconeogenesis, right? AMP and fructose 2,6 bisphosphate, you are decreasing your gluconeogenesis. So step 3 is we need to trap this glucose 6-phosphate to become glucose, right? So what is that enzyme that we need? Glucose 6-phosphatase, high yield, to convert glucose 6-phosphate into glucose. Where does this occur? Occurs mainly in your liver and your kidneys. Can other organs shunt G6P to glycogen? Yes, other organs actually shunt your G6P to your glycogen. Don't forget that glucose 6-phosphatase, it comes from the endoplasmic reticulum. All right. In terms of hormone, glucagon, remember, you're in a fasting state, it actually increases your gluconeogenesis. All right. So it increases your gluconeogenesis. It regulates your fructose 2,6 bisphosphate. Other hormones, epinephrine, cortisol, and thyroid hormone, it increases your gluconeogenesis. Think about whether you're fasting, you're stranded in a desert island, or you're exercising, or you're being chased by a bear. Anything that increases your sympathetic nervous system, right? You have increased epinephrine, increased cortisol, and thyroid hormones. You want to make more sugar, right? You are in the stress situations. You need to raise blood sugar, so you undergo gluconeogenesis. You want to break down, you want catabolic reactions, right? You want hyperglycemia as a common side effect. We're good so far with gluconeogenesis. Let's summarize, okay? This is glycolysis in reverse. What's the function of this? It can maintain blood glucose when glycogen stores are exhausted. Why? Because it must supply the brain and your RBCs, which utilize glucose for energy. Don't forget, it is not a source of energy for the liver. Why? The liver uses beta oxidation to supply the energy for gluconeogenesis. What are the substrates for gluconeogenesis? Well, it can be amino acids, except leucine and lysine, your ketogenic amino acids. Lactate can also be there, right? Glycerol 3-phosphate from your fat metabolism can be there. What else is in there? I'm blanking out. Propionyl-CoA from add chain fatty acid catabolism. Where is the pathway? What's the location? In hepatocytes, primarily in the hepatocytes. What are the non-reversible steps that must be bypassed? So first, you need to have the pyruvate carboxylase, which requires biotin and your ATP. High yield, it is also activated by acetyl-CoA. PEP carboxykinase, it requires GTP. It is activated by glucagon and cortisol. The rate limiting step in gluconeogenesis is fructose 1,6 bisphosphatase. This is activated by ATP, inhibited by AMP and fructose 2,6 bisphosphate. And then the last step, from glucose 6-phosphate to glucose, you need glucose 6-phosphatase. This is in endoplasmic reticulum of your hepatocytes. Question, what is that disease that is associated with glucose 6-phosphatase deficiency?
Von Gierke.
Excellent. So this comes from your von Gierke disease. Okay. Regulation. How am I going to stimulate this one? Remember, glucagon, acetyl-CoA, and citrate. How am I going to inhibit this? High NADH, NAD plus ratio. So gluconeogenesis is inhibited in someone who is alcoholic. That's why the pyruvate goes to lactic acid pathway. And I think that's all I can remember about gluconeogenesis. I'm sorry if I left out some important details, but I think those are the high yield points. So we're good so far with gluconeogenesis. Yep.
Yep.
Okay. So let's talk about glycogen synthesis. So from glucose 6-phosphate, we need to make glycogen. So what are the important enzymes? First, UDP glucose phosphorylase, high yield phosphorylation from glucose 1-phosphates to UDP glucose. Then glycogen synthase. This is actually the enzyme that converts the glucose into the glycogen itself. So this is high yield glycogen synthase. And you need to have a branching enzyme to make a branched version of your glycogen, because if it is unbranched, it's too long, right? It cannot be efficiently stored in your liver. So we need to make it branched to make it a compact for storage. How am I going to break down glycogen? So I'm going to break down glycogen via a debranching enzyme and glycogen phosphorylase. So put a star here, glycogen phosphorylase for breaking down glycogen. Glycogen synthase to make glycogen, and glycogen phosphorylase to break down glycogen. You can also have some enzyme in your lysosome, high yield, your alpha-1,4-glucosidase, which converts unbranched glycogen to your glucose. This is your glucose maltase, your alpha-1,4-glucosidase, which is also important in a disorder. Can you tell me what it is?
Pompe.
Come again, please.
Pompe disease.
Excellent. So this is your alpha-1,4-glucosidase. For breakdown, glycogen phosphorylase, glycogen synthase for building up. So we talk about the hormonal regulation. Methylation. When glucagon and epinephrine, you want more phosphorylase, you want more breakdown. Why? Think about sympathetics. You're running from a stressor, running from a bear, or you're fasting. You need glucose. So you need to break down that glycogen to give you glucose. If insulin is increased, remember after you've finished a meal and you're resting, insulin is increased. You want to store glucose to glycogen. That's why glycogen synthase is increased. Okay? So when insulin is ruling, glycogen synthase is increased. You're storing glycogen to glucose. When glucagon and epinephrine is ruling, glycogen phosphorylase is increased. You want to increase the conversion of glycogen into glucose. Another one here is a schematic diagram of your epinephrine and your glucagon. Remember, epinephrine and glucagon, they both increase your cyclic AMP. They both increase your pKa. They increases your glycogen phosphorylase for glycogen breakdown. Again, on the other hand, they decrease your glycogen synthase. They inhibit glycogen synthesis. We talk about the regulation. This is just a nice slide for the regulation of glycogen. All right, let's talk about the glycogen storage diseases, your favorite. So von Gierke's disease, this is glucose 6 phosphatase type 1, presents in infancy, severe hypoglycemia. Remember, you cannot make glucose. Glucose 6 phosphate cannot become glucose. What can we see in a patient, in a neonate? Lethargy, seizures, and lactic acidosis, right? By the Cori cycle. You can also have enlarged liver, hepatomegaly, which can lead to liver failure. How are we going to diagnose? Biopsy before, but now you do DNA testing, right? Treatment, cornstarch. Give cornstarch. It's a glucose polymer. However, you avoid sucrose, lactose, fructose, your simple sugars, and your galactose. Why? Because this one, they feed into the glycolytic pathway. And remember, it worsens accumulation of glucose 6 phosphate. You cannot convert glucose 6 phosphate to glucose, okay? Because you cannot metabolize glucose via gluconeogenesis because you don't have glucose 6 phosphatase. Pompe's disease type 2. This is your lysosomal acid maltase from your branched amino acid to glucose. You can have this one in your lysosomes. So you have accumulation of glycogen in your lysosomes. Classic form in infancy. This is very severe, often death in infancy. What can we see? Enlarged pumps. When you think Pompe, think about pumps. So you have cardiomegaly, enlarged heart. You can have enlarged tongue. You can have hypotonia, enlarged liver, hepatomegaly. And the most common cause of death in Pompe is heart failure. Cori's disease, on the other hand, is your glycogen storage disease type 3. This is a debranching enzyme. Similar to von Gierke's, except milder hypoglycemia, no lactic acidosis. Remember, Cori cycle is still intact in your liver. And you have muscle involvement. You don't have muscle involvement in the Cori's disease. What's the key point here in Cori's? Gluconeogenesis is intact. Contrast this one with von Gierke's, where gluconeogenesis is kaput. It's gone. You don't have glucose 6 phosphatase there. So let's talk about the Cori cycle. What is this? The Cori cycle, or the lactate cycle. So in the muscle, glucose is converted to pyruvate, and then the last one is the lactate. So if you're exercising, right, you're producing much more lactate in the muscle. How am I going to get rid of this muscle? How am I going to convert this lactate into glucose? So this lactate from the muscle, it goes back to the liver. It's acted up, it becomes pyruvate, and then you can have glucose. So this is the Cori cycle. The lactate coming from the muscle can be converted back to glucose in our liver, because our body wants to conserve stuff, right? We don't just want to waste stuff. So from lactic acid to glucose. So if you have type 3, what's the presentation? Hypoglycemia, hepatomegaly. You can have hypotonia or weakness, possible cardiomyopathy with hypertrophy. But I'm thinking hypotonia, weakness, and hypoglycemia and hepatomegaly for Cori's. How about McArdle? Muscle glycogen phosphorylase. So a myophosphorylase deficiency. Why? This enzyme breaks down glycogen in your muscle cells to provide glucose for your muscles. In this disorder, you have a deficiency in a myophosphorylase. So what can we see? It is in adolescence and early adulthood. Contrast this one with your first 3 disorders, which is much more common in infancy and the neonatal period. So what can we see? Fatigue and cramps after physical activity. In the vignette, we're going to say the patient has cramps, fatigue, exercise intolerance. Think about McArdle's. If you look at the lab, you have myoglobinuria from the breakdown of the muscles, and you have a CK release with exercise. Remember, why do we have myoglobinuria and creatinine kinase release in the muscle? Since you cannot break down glycogen in the muscle turns to protein catabolism to provide energy. That's why you have myoglobinuria and CK and creatinine kinase release. Okay, it was not supposed to happen, right? Because the preferred energy for the muscle is glucose, right? Here in McArdle's, the urine may turn dark after exercise because you are catabolizing your proteins in your muscles, which are not supposed to happen. Again, glucose to pyruvate. This is your glycolysis pathway. Don't forget, glucose 6-phosphate can be shunted. It can become a substrate of your HMP shunt. Let's go back to term 1. What is HMP shunt?
Hexose monophosphate shunt, or the PPP.
So what is the function of HMP shunt? Why do we need HMP shunt?
NADPH.
Excellent, NADPH, right? From glucose 6-phosphate to fructose 6-phosphate. We are producing NADPH. Why is this important in red blood cells?
Reactive oxygen species.
How? Can someone tell me how? How does it reduce the production of reactive oxygen species?
With glutathione reduction.
So remember, it produces reduced glutathione, right? Via the enzyme glutathione reductase. So you produce reduced glutathione to react with this H2O2 to make H2O and oxygen, right? And then you have the glutathione peroxidase to bring it back, to oxidize glutathione. Main point here, NADPH is used for the production of reduced glutathione in order to react with H2O2 to make H2O and O2. Okay, that's good. So preventing reactive oxygen species in your red blood cells. Good job. So NADH, and again, ribose 5-phosphate for nucleotide synthesis. Can someone indulge me? How am I going to make your nucleotides? Let's see, purine synthesis. From ribose 5-phosphate to what? Purine synthesis.
Is it PRPP?
Excellent. From PRPP to what?
To IMP.
Excellent. From IMP to what? GMP and AMP. Can you give me a drug that inhibits conversion of IMP to GMP? Your mycophenolic acid, don't forget that one, which inhibits IMP dehydrogenase. And of course, if you have Hep C, ribavirin. How about pyrimidine synthesis? Can someone tell me about pyrimidine synthesis?
The rate limiting enzyme is CPS2.
Excellent, CPS2. And then to convert what? Orotic acid, from orotic acid, remember to UMP, right? Are we clear there? From UMP to DUMP, and then DTMP is the end, right? Please revise your purine and pyrimidine synthesis. You have orotic aciduria here in your pyrimidine synthesis. And my question is, how am I going to differentiate megaloblastic anemia from orotic aciduria, from folate deficiency, and from vitamin B12 deficiency? So you have orotic aciduria to those three diseases. How do I know that it is due to folate deficiency, or it is due to B12 deficiency, or just due to defect in pyrimidine synthesis? That's my question.
I think with B12 deficiency, you see neurological symptoms.
Good job. What else? So just give them a supplement. Just give them B9 and B12. Just give them a folate supplement or a B12 supplement. If there's improvement, then it is that deficiency. But if you give them B9, no improvement, B12, right? No improvement, then it is a defect in pyrimidine synthesis. That's what I'm getting at, because the three of them has orotic aciduria, okay? So we talk about G6PD deficiency and thiamine deficiency for trans-ketolase. How am I going to detect thiamine deficiency? The easiest way to detect thiamine deficiency, a test.
Blood sample.
Excellent, blood draw. So you just do a trans-ketolase assay. Look at the trans-ketolase activity in the blood. And then you can say if they have a thiamine deficiency. That's actually high yield. Okay, so there are other sugars that needs to be converted, right? So fructose. Let's talk about fructose metabolism. First, fructose is converted to fructose 1-phosphate via fructokinase. And then fructose 1-phosphate to dihydroxyacetone phosphate. Remember, dihydroxyacetone phosphate can be converted to glyceraldehyde 3-phosphate, and then they will get shunt to your glycolysis pathway, okay, to make pyruvate. Now, the high yield point here is this: fructokinase deficiency and aldolase B. Which one is kinder, fructokinase deficiency or aldolase B?
Fructokinase.
Fructokinase deficiency. So anything that has worse symptoms like hepatomegaly, you can have aldolase B deficiency. So fructokinase is kinder than your aldolase B. So, as I've said, this is benign essential fructosuria, deficiency of fructokinase. Fructose not taken up by liver cells, and they appear in the urine. Contrast that with your hereditary fructose intolerance. So the baby has just weaned from breast milk. You have failure to thrive. You have hypoglycemia after feeding. You have seizures. They can have enlarged liver. Treatment, you need to avoid fructose, sucrose, or sorbitol. Again, glucose can be converted to fructose. in different parts of our body. We call this one the Polyol Pathway. So glucose can be converted to sorbitol via aldose reductase, and sorbitol to fructose via sorbitol dehydrogenase. However, in the eye, there is a deficient enzyme. What is it? In the eye, you have a deficient enzyme.
The sorbitol dehydrogenase?
Excellent, the sorbitol dehydrogenase. You don't have sorbitol dehydrogenase in your eye, therefore sorbitol cannot be converted to fructose. That's why if you're a diabetic, you have an increase in glucose, you have an increased activity of aldose reductase, you have increase in sorbitol. In your eye, it accumulates there. Okay, so you have increased intraocular pressure in your eye, so you have clouding of your lenses, you cannot see clearly. Okay, why? You don't have sorbitol dehydrogenase in your eyes to convert sorbitol to fructose. In diabetics, which is increased aldose reductase, which converts glucose to sorbitol. Those two enzymes are high yield. How about in terms of galactose? So galactose can be converted to galactose 1-phosphate via galactokinase, and galactose 1-phosphate to glucose 1-phosphate via galactose 1-phosphate uridyl transferase. That's a mouthful, so let's just call it GALT. Which one is kinder? Galactokinase. Which one is worse? GALT deficiency. So classic galactosemia, this is your GALT, autosomal recessive. Galactose accumulate in the cell, so it leads to accumulation of galactitol in your cell. Galactokinase deficiency, milder form of galactosemia, it accumulates in the blood and urine. High yield, you can only see cataracts in your galactose metabolism disorders. Cataracts only in galactose metabolism disorders. So you have may present as vision problems. Look here at the lens of the eye of the child. You can see galactitol accumulating in the lens. Okay, we're good so far? So what's common between your fructose and galactose metabolism disorders? What is common to them?
Reducing sugars in the urine.
Excellent. So you have a reducing sugar in the urine. How do we contrast them? If they have cataracts. If in the vignette there's a cataract, think about galactose metabolism. Which one is kinder? Always the first enzyme is kinder. In fructose, it's fructokinase. You have mild symptoms. In galactose, it's the galactokinase deficiency, which has some milder symptoms. The worst symptoms comes from the deficiency of the second enzyme. For fructose, aldolase B. For galactose, you have gout. Okay, good job. Now, pyruvate dehydrogenase complex, to convert this one to acetyl-CoA. You can also make pyruvate to go back to glucose, make this back to glucose via gluconeogenesis. What's the enzyme? Pyruvate carboxylase. Now, from pyruvate to acetyl-CoA, I need a pyruvate dehydrogenase complexes. Now, there's an important cofactors here. Can you give me the five or four coenzymes for your pyruvate dehydrogenase complex? I think 5.
5.
Yep, could you tell me that?
TPP, lipoic acid, FAD, NAD, and CoA.
Excellent. So these are the lovely coenzyme for your
Tender loving care for Nancy, as Dr. Mai likes to remind us.
okay, that's also a good one, right? So you need to have those. So another one, dehydrogenase, your TLC-FN. So any dehydrogenase in the TCA cycle, they also need those 5 coenzymes, okay? Don't forget that. When you think dehydrogenase, thiamine, it's all in there. And the 5, any dehydrogenases in the TCA cycle also needs those 5 coenzymes. Okay, here they are, the cofactors, I'm sorry. So NAD, FAD, coenzyme A, thiamine, and lipoic acid. Again, let's go to the TCA cycle. Now you convert your pyruvate to acetyl-CoA. We're going to go to the TCA cycle. Memorize the substrate. They are important. But the question will going to be regulation, always about regulation. What's the end point of your TCA cycle? Oxaloacetate. That's the end point. If we join acetyl-CoA and oxaloacetate, we make citrate. That's a beginning point. And we have intermediate substrates here. I want to highlight alpha-ketoglutarate, succinyl-CoA, and you have the fumarate. So from isocitrate to alpha-ketoglutarate, you are removing CO2 and NADH. You are converting NAD plus to NADH. From alpha-ketoglutarate to succinyl-CoA, you are also removing CO2. You are converting NAD plus to NADH. Succinyl-CoA to succinyl GTP is involved. You are making GTP. Okay, so from GDP to GTP here. Succinate to fumarate, FADH2. This is where the FADH2 comes from to go to your ETC. From malate to oxalate, there's also an NADH molecule there. But the questions will going to be about regulation. Citrate can go to fatty acid metabolism. Alpha-ketoglutarate can go to amino acid metabolism. Oxaloacetate can be converted back to glucose and to amino acid. So TCA cycle is important because it can provide substrate to different pathways. It can provide substrate for fatty acids, amino acids. It can be directly converted to glucose via gluconeogenesis. So this pathway is very, very important. So now let's look at the high yield parts. So you have oxaloacetate plus acetyl-CoA. What's the enzyme? Citrate synthase to make citrate. Isocitrate and alpha-ketoglutarate, isocitrate dehydrogenase. Alpha-ketoglutarate to succinyl-CoA, you have the alpha-ketoglutarate dehydrogenase. Since they are both dehydrogenases, they both need the 5 cofactors, right? As I've said again, if you have an increased energy, your molecule, you have an high energy molecule like ATP and NADH, you don't need the TCA cycle. So any high energy molecule like citrate and ATP, it inhibits your citrate synthase, your isocitrate dehydrogenase, your alpha-ketoglutarate dehydrogenase. What's the rate limiting step here? Isocitrate dehydrogenase. As I've said before, low energy molecules, if there's a low energy molecule like ADP and calcium, you need to stimulate the TCA cycle. You need to make more of the intermediates of your TCA cycle. So low energy molecules like ADP and calcium, high yield, they activate your isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase to make your TCA cycle more active. We're good so far with your TCA cycle. So know the intermediates, know the enzyme that's involved. All right, so let's summarize, okay? Is that okay with everyone? Let's just summarize. So TCA cycle, what's the function? The other name, Krebs, what's the function? So you generate high amounts of NADH, here, NADH, 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 and what? FADH2. So what if I generate those molecules? So that they can act as fuel for your ATP synthesis in the electron transport chain. Where does this occur, the TCA cycle? In the mitochondria. Does it require oxygen to function? Of course, it requires oxygen to function. What's the equation? Acetyl-CoA plus 3 NAD plus FAD plus GDP plus inorganic phosphate. You have 2 CO2, 3 NADH, you have CoA, you have GTP, and you have FADH2. Okay, so how many ATPs can the TCA cycle yield? Approximately, in theory, it can yield 12, 12. It can yield 12 ATPs if the ETC were 100% efficient. There's a caveat there. Okay, so don't forget, both carbons of acetyl-CoA leaves a CO2 in two reactions here. In your dehydrogenase reactions, you have a decarboxylation here. You're removing CO2. Important enzymes, let's highlight. Isocitrate dehydrogenases is stimulated by decrease in energy, inhibited by increase in energy. Another important enzyme, alpha-ketoglutarate dehydrogenase. It requires the same cofactors as pyruvate dehydrogenase complex. Don't forget B1, B2, B3, B5, and your lipoic acid. In alcoholics, B1 deficiency leads to Wernicke's encephalopathy. You have ataxia, confusion, and ophthalmoplegia. This enzyme is inhibited by increase in energy, like ATP or NADH. It is also inhibited by intermediates in the cycle. If you have an increase in succinyl-CoA, right, you don't need this enzyme. Another one, succinyl-CoA synthase here, succinyl-CoA synthase, it generates GTP. So the enzyme here from succinyl-CoA to succinate is succinyl-CoA synthase. So an important intermediate, I think, here is citrate. It functions to shuttle acetyl-CoA out of mitochondria for your fatty acid synthesis. Don't forget the citrate shuttle for synthesis of fatty acids. Succinyl-CoA also, where's the succinyl-CoA for having a mental block, for heme synthesis. Sorry. Succinyl-CoA is for heme synthesis. Another one is fumarate. Fumarate is for, yes, urea cycle. It enters the urea cycle. Malate, gluconeogenic. Okay, gluconeogenic substrate. It is a substrate for gluconeogenesis. Is the TCA cycle regulated by hormones? Yes or no? What do you think?
No.
No. So what is it? What's the regulation of the TCA cycle? Primarily.
Substrate availability.
Exactly. Energy status. If it's high energy, if you have increase in high energy molecules or lower energy molecules. Please check what I've said about the TCA cycle. So most of these are from my memory. So sometimes I don't trust my memory. So please double check. Okay. Now let's go to electron transport complex. So you have complex 1, complex 2, complex 3, complex 4. We are in the mitochondria. We have the cytosol there. This is the outer membrane of the mitochondria. We have the intermembrane space. This is where the H+ we're going to go. And we have the inner membrane. So we have now NADH. Remember the NADH from the TCA cycle. Where does it go? Complex 1. So complex 1 here, we're producing H+. The electron is transferred by CoQ to complex 3. Okay. So complex 1, as I remove the H+ in my NADH, as I oxidize, right? Oxidation is loss of electrons. So you're removing H+. So as I oxidize my NADH, the H+ we're going to go in the intermembrane space. And what happens to the electrons? It is transferred to complex 3 by your coenzyme Q. And then What happens? From complex 3 again, there's an H plus. The electrons is transferred to complex 4 via your cytochrome C. Throughout the electron transport chain, the H plus is always in the intermembrane space. It is increased in this space. Okay, where are the complexes? They are in the inner membrane of the mitochondria. Remember, the mitochondria has 2 membranes, outer membrane and the inner membrane. So cytochrome C from complex 3 to complex 4. So here it is. NADH, complex 1, CoQ transferred to complex 3. From complex 3, the electrons will going to transfer to complex 4 via cytochrome C. As we go over this electron transfer, the H plus is in your intermembrane space, still building up there. So building up there. Why does it build up there? We need this one as our fuel for complex 5, for our ATP synthase. The end acceptor is your oxygen, which makes water. Okay. The acceptor in the electron transport, the last acceptor is your oxygen to make water. So now you have too much H plus in the intermembrane space. What are we going to do? We need this one to drive the motor of your ATP synthase to make ATP, to convert ADP to ATP. The H plus, it provides an electromotive force, right? This is positively charged. It drives the motor of your ATP synthase to make more ATP. High yield, what are the inhibitors? Rotenone, complex 1. 1 rotenone, 1 binds to complex 1. Antimycin and 3 mycin, complex 3. 4, complex 4, you have, it ends in ide, monoxide, cyanide. So if it ends in ide, it inhibits complex 4. Now, contrast your inhibitors with your uncoupling agents. So uncoupling agents, they disturb your inner membrane. So you don't have the increased concentration of H plus in your intermembrane space because they poke holes there. So you cannot make ATP. You don't have the force to drive the motor of your ATP synthase to make ADP to ATP. We're good so far. How about FADH? Where does it go? What complex does it go? Here, where does it go?
It is by
Complex 2.
Complex DOS. Excellent. So complex 2. So NADH is complex 1. FADH is complex 2. And then we're going to go to 3 and then 4. Final electron acceptor, oxygen to make what? Water. You need to know the inhibitors. Uncoupling agents, they put holes. Can you give an example of uncoupling agents?
Aspirin.
Aspirin, reversible or irreversible inhibitor of COX?
irreversible.
Irreversible inhibitor of your COX. High Km, low Km, same Km.
Same Km.
Excellent, same Km. What happens to Vmax though?
No Vmax.
Excellent, decrease in Vmax. We're integrating materials. Do it now. Okay, integrate stuff. So let's summarize. Okay, let's summarize the electron transport chain. What's the function? Those NADH and FADH2 from your TCA cycle, they need to become ATP. So this mechanism, your electron transport chain, they convert NADH and FADH to ATP synthesis. What's the process called? Oxidative phosphorylation. So 3 ATP per NADH and those 2 ATPs per FADH2. FADH2 has a lower energy content than NADH. ETC is located in inner mitochondrial membrane. You have NADH and FADH2. They create a proton gradient in your inner membrane. They pass electrons in a stepwise fashion. Oxygen is the final electron acceptor, and the flow of protons in your intermembrane space back down the concentration gradient. They drive your F0 and F1 ATP synthase complex. What's the clinical importance of your electron transport chain? So your electron transport inhibitors, they disrupt the membrane-bound carriers, resulting in decreased proton gradient, decreased ATP synthesis, decreased oxygen consumption, also carbon monoxide poisoning, right? Complex 3 inhibitors. Also cyanide poisoning. Complex 3 inhibitor. Complex 3 or complex 4? Complex 4, I'm sorry. Because complex 3 is antinomycin, right? So complex 4, your IDEs. So don't forget carbon monoxide poisoning and cyanide poisoning. For ATPase inhibitors, there's also an inhibitor for your ATP synthase that is high yield. We call this one oligomycin. Oligomycin inhibitor of your ATP synthase. So meaning you have increased proton gradient in the intermembrane space. However, you cannot make ATP because the electron transport chain stops. That's your oligomycin. For uncoupling agents, they increase the permeability of the membrane, resulting in what? Decreased proton gradient in the intermembrane space. You have increased oxygen and NADH consumption. The ATP synthesis stops. However, the electron transport chain continues, thereby generating heat. Right? That's why if you're into losing weight, if you're taking dinitrophenol, 2,4-dinitrophenol, which is thermogenic, or if you're taking aspirin, or if you have in kids, they have thermogenin in brown fat. They are uncoupling agents. The ATP synthesis stops. However, the electron transport chain continues, which produces heat. Okay, I think that's the high yield point in your high yield points in your electron transport chain. We're good so far. We're good?
Oh, yes.
Okay. Fatty acid synthesis, citrate to cytosol via the citrate shuttle. So we are in the mitochondria now. We need to get out this citrate to synthesize fatty acids. What's the point here? Acetyl-CoA cannot be converted. It cannot cross the membrane, right? So it needs to be paired with oxaloacetate to make citrate. And then citrate can get out of the mitochondria, okay, via the citrate cycle. And then citrate can be converted to acetyl-CoA via ATP citrate lyase. However, that's not the most important thing. The most important thing is this, acetyl-CoA carboxylase, where acetyl-CoA is converted to malonyl-CoA. Don't forget carboxylase, it needs biotin, right? B7. What increases fatty acid synthesis? Insulin, don't forget, insulin anabolic reactions. When insulin is ruling, think about anabolic reactions. Or citrate from your TCA cycle. You need to make fatty acid. If you're in the fasting state, glucagon and epinephrine. Think about fight or flight. Think about starvation, dieting, right? You inhibit acetyl-CoA carboxylase. You don't want fatty acid synthesis. You need oxidation. You need to break down fatty acids. Malonyl-CoA can be broken down via the process of beta oxidation. So malonyl-CoA is an important inhibitor of fatty acid breakdown. We're gonna look at that next couple of slides. What's the end point of fatty acid synthesis? Palmitate. We're gonna make 16 carbon fatty acid. What's the enzyme for this? Fatty acid synthase. It requires NADPH from your HMP shunt. Now, if citrate shuttle for making fatty acids, how about we break down fatty acids? When glucagon or epinephrine is ruling, what happens? So you have your triacyl glycerol molecule, you have the glycerol head here, and you have the 3 fatty acids there. You have HSL, hormone sensitive lipase. To break it down to glycerol, you remove the head, you retain the fatty acid. And then this fatty acid can be converted to fatty acyl-CoA via long chain fatty acyl-CoA synthetase. So you need ATP, you also need CoA. Don't forget, fatty acyl-CoA synthetase to make fatty acyl-CoA. Then this fatty acyl-CoA, we're going to go to your mitochondria via carnitine shuttle. Carnitine shuttle. Citrate shuttle synthesize fatty acids. Citrate synthesize. Carnitine shuttle, think about carnage. So you're breaking down fatty acids. Okay? Don't forget, here it is, your carnitine shuttle. The malonyl-CoA, if malonyl-CoA is increased, it inhibits your breakdown of your fatty acids. So fatty acyl-CoA from the cytosol, we're going to travel back to the inner membrane space. It will going to be converted to acyl carnitine. If we wanted to make more carnitine to make fatty acyl-CoA. So in the cytosol, fatty acyl-CoA, it goes back to the inner membrane space. This is now your fatty acyl-CoA. There's an enzyme that regulates the conversion of the fatty acyl-CoA to acyl carnitine. We call this one CPT1. This is high yield. This is the enzyme that is inhibited by malonyl-CoA. So CPT1 is only active in breaking down of your fatty acids. So the questions were going to be clinical. What happens if you have primary systemic carnitine deficiency? So you have a mutation that affects carnitine uptake. What can you see? Everything is enlarged. Hepatomegaly, encephalopathy. You have hyperammonemia. Right? You have low serum carnitine. You have hypoketotic hypoglycemia. You have a decrease in blood sugar. However, you have no increase in ketones. Remember, the normal response is prolonged starvation, right? You have ketogenesis. You have ketosis, You have reproduction of your ketones. However, in this case, in primary systemic carnitine deficiency, you are hypoglycemic. However, the ketones are not elevated. So we call that hypoketotic hypoglycemia. Reduced carnitine levels in everything, in muscle, liver, and the heart. Contrast that with MCAD deficiency. MCAD is, you have a medium chain acyl-CoA dehydrogenase deficiency. It's autosomal recessive. It is also ketotic hypoglycemia. However, this is severe hypoglycemia without ketones. What can we see in the blood? Dicarboxylic acids in the urine, and high acyl carnitine levels in the blood. Okay, so how are we going high yield to contrast primary systematic carnitine deficiency and MCAD deficiency? Only in MCAD deficiency can you have dicarboxylic acid in the urine and high acyl carnitine levels in the blood. That's high yield. Again, propionyl CoA. We're talking about propionyl CoA. Why? Because most amino acids can be converted to propionyl CoA. Cholesterol and odd chain fatty acids is converted to propionyl CoA to become succinyl CoA so that it can be metabolized in your TCA cycle. It can be used as a substrate for your TCA cycle. So don't forget that propionyl CoA is converted to methylmalonyl CoA, and this methylmalonyl CoA is converted to succinyl CoA. Via an important cofactor, you need B12. So you have methylmalonyl CoA mutase to convert it to succinyl CoA. It needs B12. It also needs biotin to convert it from propionyl CoA to methylmalonyl CoA. Why is propionyl CoA important? It's a common pathway to TCA cycle. Look, amino acid metabolism, cholesterol, odd chain fatty acid metabolism, all converted to propionyl CoA. And this propionyl CoA is converted to succinyl CoA. Why? So that it can be used as a substrate in the TCA cycle. An elevation of methylmalonyl CoA is seen in B12 deficiency. How about if I'm fasting or you're dieting? What happens? You have an increased production of ketone bodies. This is alternative fuel source for some cells due to fasting or starvation. So the acetyl-CoA is shunted towards the ketone bodies. So we have breakdown of fatty acids to acetyl-CoA. Instead of acetyl-CoA going into TCA cycle, it goes to production of your ketone bodies. Why? The increase in acetyl-CoA, it's too much. It exceeds the capacity of your TCA cycle. Voila, this is the ketone body synthesis. Do I need to memorize everything? No. The most important thing is this. The end product is acetone and 3-hydroxybutyrate. So the beginning point is acetyl-CoA and then HMG-CoA. There's an enzyme here, HMG-CoA reductase, which is important in cholesterol, in controlling cholesterol. So if you're prescribed with statin drugs, your atorvastatin, anything that ends in statin, it inhibits HMG-CoA reductase. You are reducing cholesterol, de novo cholesterol synthesis. Because remember, even though you're not eating egg yolks, right, your body can synthesize cholesterol. So the end point here is acetone and 3-hydroxybutyrate. How can we detect acetone? In the breath. How can we detect hydroxybutyrate? Check in the urine. You can have ketone bodies in your urine. Ketolysis. What is this? The 3-hydroxybutyrate here, the end product of your ketone body synthesis, can be converted to ATP. So the liver releases ketones into the plasma. Okay, it is used by muscle and your heart. Why? Because the glucose is prioritized for the brain. The brain loves glucose, right? However, if it's prolonged starvation, the brain can use ketone bodies, but the liver cannot use ketone bodies. High yield, the brain can use ketone bodies, the liver cannot use ketone bodies. So what's the big picture here? The brain cannot use fatty acids, so the ketone bodies allow the use of fatty acid energy by the brain. It is also used by other tissues, so to preserve glucose for the brain. Okay, that's the big picture here. Let's talk about diabetes. What is diabetes? So low insulin, high fatty acid utilization. You have oxaloacetate is depleted in the TCA. The TCA cycle stalls. Why? You no longer have a substrate, oxaloacetate. Remember, oxaloacetate plus acetyl-CoA makes citrate, which is the beginning product of your TCA cycle. So that the cycle we're going to start, right? You have increase in acetyl-CoA and then you have ketone production. The acetyl-CoA cannot go to the TCA, right? So it goes to ketogenesis, the formation of more ketones. Okay, guys, alcoholics, right? So if you're a chronic alcoholic, what happens? You have an excess in NADH. So your oxaloacetate cannot be shunted to malate in your TCA cycle. Therefore, it stalls your TCA cycle. Since TCA cycle is stalled, you have more and more substrate of acetyl-CoA. It cannot go into the TCA cycle. So what happens? Well, it can go via ketogenesis. You increase production of ketones. Or it can also decreases gluconeogenesis. Why does it decrease gluconeogenesis? Because you are removing NAD+. You are increasing NADH. In terms of the urinary ketones, normally there's nothing in the urine. So if you have urinary ketones high yield, what does it mean? Either you have an uncontrolled diet, BDs, or you're suffering from DKA, especially for type 1, or you're starving, prolonged starvation, or alcoholics. If you've gone binge drinking, it can increase your urinary ketones. Again, high yield, put a star here, ethanol metabolism. So for ethanol metabolism, ethanol is converted to acetaldehyde via alcohol dehydrogenase. Remember, NAD+, we need that NAD plus. So if you deplete NAD+ in alcoholics, right, you cannot undergo glycolysis. So you can decrease gluconeogenesis. All of that, all of your acetyl-CoA will gonna go with ketone production. All of the pyruvate can go to lactic acid production. From acetaldehyde, it goes to mitochondria and converted to acetate via aldehyde dehydrogenase. High yield, put a star here, alcohol dehydrogenase and aldehyde dehydrogenase. Fomepizole inhibits alcohol dehydrogenase, commonly used in overdose alcohol and ethanol poisoning, right? Fomepizole. And you have aldehyde dehydrogenase, you have a disulfiram, which is not commonly used in chronic alcoholics to prevent you from drinking alcohol. Okay, I think that's the most important points there. As I've said, alcohol dehydrogenase is there. Treatment for methanol and ethylene glycol intoxication. Aldehyde dehydrogenase, disulfiram, this is the antabuse. What happens if aldehyde dehydrogenase is gone? Acetaldehyde accumulates, so it triggers catecholamine release. You have sweating, flushing, palpitations, nausea, and vomiting. What is alcohol flushing? Commonly seen in when consuming alcohol. Us Asians, it is very common among our population. Among Asians, you have Japanese, Chinese, Korean. You have an inherited deficiency in aldehyde dehydrogenase 2, ALDH2. Is this bad? Well, there's some study that says it increases the risk for esophageal and oropharyngeal cancer, specifically squamous cell carcinoma of the head and neck. More of that in term 4. Now, ethanol metabolism, if I go to breweries, right, if I binge alcohol, what happens? It can lead to all kinds of problems. Hypoglycemia, right? Why? Decreased gluconeogenesis. Ketone body formation, ketosis. Why? TCA cycle stalls. I have increased acetyl-CoA. The increased acetyl-CoA will gonna go to ketone formation. Lactic acidosis. Why? Pyruvate not be converted to pyruvate cannot be used in the TCA cycle, so it will gonna be metabolized by your lactate dehydrogenase, thereby producing lactic acids. We talk about accumulation of fatty acids. You can also develop hepatitis and cirrhosis. High yield. What's the trigger for all of this? Why do we have all of this? What's the biochemical trigger? Increase in NADH. So the ratio of NADH and NAD plus is increased. Okay. Don't forget, NADH, it stalls the TCA cycle. It's a high energy molecule. That's why you have ketone production and you have decrease in gluconeogenesis and hypoglycemia. I explained that in the previous slide. So what happens for the ketones from your acetate? Of course, acetyl-CoA can be converted into ketones. From acetate to acetyl-CoA, since TCA cycle is not working due to high NADH, the acetyl-CoA can be converted into your ketones. I've talked about lactic acidosis. Why do you have lactic acidosis? I've explained that. You have increase in pyruvate, which is shunted to lactate. You also have accumulation of fatty acids. Why? NADH. Everything is related to NADH. It stops beta oxidation. Therefore, you have decrease in fatty acid breakdown. What are the effects of insulin? Remember insulin, think about anabolic, building up reactions. Synthesis, think about glycogen synthesis. Think about glycolysis, you're producing ATP. It promotes glucose to adipose tissue to form triglycerides. You are synthesizing fat. It promotes uptake of amino acids by muscle. You are synthesizing protein. It stimulates protein synthesis. Insulin, think about anabolic reactions. You're building up stuff in your body. Contrast that with glucagon. Glucagon, think about catabolic reactions. Everything is being broken down. So glycogen is broken down, glycogenolysis in your liver. What else? Fatty acid breakdown, increase in fatty acid breakdown. What else? You have your decrease in triglycerides, your increase in hormone-sensitive lipase. We need to break down those fat, right? What else? It stimulates gluconeogenesis. Glucagon, think about catabolic reactions. You're breaking down stuff. Again, this is a high-yield slide. So first is what happens? Your glucose sources. So you eat, right? After a couple of hours, it's gone. Your ingested glucose is gone. Within the first 24 hours, your energy sources comes from glycogen, right? From glycogen, and then after 24 hours, it comes from other sources. So the point here is this. Glycogen only gives you energy within 24 hours after you last eat a meal. So it needs 24 hours. All of that time, your body is being provided by glycogen, right? Glycogen is being broken down to become glucose. After 24 hours, your body tries to get other sources. The glycogen stores in your liver is depleted, right? And you're still starving. So what happens? Can I break down muscles? I don't want to do that. That's the last resort. So what can I do first? Break down fat. I need to increase my hormone-sensitive lipase to break down fat. The last resort, if I cannot do that efficiently, is muscles. Our body hates to degrade amino acid. That's the last resort, okay? Because why? Remember, urea is toxic to the brain. Okay, so that's the key point here. Glycogen exhausted after 24 hours, and the glucose levels are maintained in fasting by many sources, specifically your fat. Okay, the last resort is protein catabolism. Again, malnutrition, kwashiorkor. Kwashiorkor is inadequate protein intake. So we have a decrease in albumin in the blood. What happens to the oncotic pressure and what happens to the hydrostatic pressure?
The oncotic pressure decreases and the hydrostatic increases.
Excellent. So the oncotic pressure decreases and hydrostatic pressure increases. So hypoalbuminemia, it can cause edema. So you can have swollen legs and abdomen. Now, contrast this one with marasmus. Marasmus is inadequate energy intake. So you are You have insufficient in total calories. You don't have edema. You have muscle and fat wasting. MSUD, maple syrup urine disease. This is a branch chain amino acid disorder. What's the enzyme that's deficient? Alpha keto acid dehydrogenase. Don't forget, it's a dehydrogenase, so it needs your enzymes, your thiamine, your cofactors, thiamine, and your lipoic acid, those 5. Amino acids and alpha keto acids in the plasma and the urine. So the urine smells sweet. They're going to tell you in the vignette. Why? Because of the isoleucine. The isoleucine, which is alpha keto acid, it gives the urine the sweet smell. So what amino acids cannot be given in someone who has maple syrup urine disease?
Isoleucine, leucine, and valine.
Excellent. So you have I love Vermont, right? Isoleucine, leucine, and valine. Excellent. Now let's talk about urea cycle disorders. So this one is the onset is in newborn. So after feeding, you have a protein load which produces symptoms. Why is this deadly? It can lead to seizures. So what can we see? Severe hyperammonemia. Okay, so in the blood, can you imagine 1,000 micrograms in a newborn per deciliter, right? For this one. So you have hyperammonemia. In the vignette, if there is hyperammonemia, think about urea cycle disorders. OTC deficiency, most common urea cycle disorder. This is increase in carbamol phosphate, which increases the orotic acids. Again, OTC deficiency, most common. Why? You have an increase in carbamol phosphate here. You have an increase in orotic acids. Again, CPS2 and CPS1, right? So there's a concept called glucogenic and ketogenic amino acids. Ketogenic amino acids, there's only 2. What are those?
Leucine, lysine.
Yes, that's it. Everything else is glucogenic. What does it mean if it's glucogenic and ketogenic? Ketogenic amino acids can be converted to ketone bodies and acetyl-CoA. They cannot become glucose. Okay, all of your glucogenic, they can be converted to pyruvate or your TCA cycle to become intermediates, right? They can become glucose via gluconeogenesis. So that's it. Using lysine, ketogenic amino acids, they cannot become glucose. They can only be converted to ketone bodies and acetyl-CoA. PKU, phenylketonuria. Can we do the catecholamine synthesis from term 1? So we have phenylalanine to tyrosine. What's the enzyme?
Phenylalanine hydroxylase.
What's the cofactor?
BH4.
Excellent. Remember, if you see hydroxylase, think about tetrahydrobiopterin. So from tyrosine to L-DOPA,
Tyrosine dehydrogenase.
tyrosine,
Dehydrogenase.
is it dehydrogenase or hydroxylase? Hydroxylase, yes, excellent. Tyrosine hydroxylase. And then from L-DOPA to dopamine,
Dopa decarboxylase.
excellent, dopa decarboxylase. Dopamine to norepinephrine.
Dopamine beta hydroxylase,
Dopamine beta hydroxylase. What's the cofactor there? The important cofactor.
vitamin C,
Excellent. And then from norepinephrine to epinephrine.
PNMT,
What's the cofactor? What's the important cofactor?
SAM.
SAM. Good job. There you go. How are we going to Break down catecholamine, norepinephrine and epinephrine. What are the two enzymes?
COA.
MAO and COMT.
MAO and COMT. So PKU, this is deficiency of phenylalanine hydroxylase. Thank you for that. Well done. So defective enzyme, this is a classic. Or you have a deficient in BH4. You have accumulation of phenylalanine. So what is essential here is you give tyrosine. Tyrosine becomes essential in PKU. A rare defect is BH4 deficiency. Okay, don't forget that one. Voila, there it is. Tyrosine, dopa, dopamine, norepinephrine, norepinephrine 2, epinephrine. How are we going to break it down? Don't forget, tyrosine can be converted to fumarate. And fumarate is a substrate of your TCA cycle. Now, what is that? Enzyme deficiency in homogenesic acid. You have an increase in homogenesic acid. You have a dark urine.
Alkaptonuria,
Excellent. Alkaptonuria.
homogenesic acid, oxidase, yeah.
Yes. So you have a deficiency in homogenesic acid oxidase. You have an increase in homogenesic acid. It deposits into your joints. So you can have darkened urine. You can have dark
Connective tissue.
Yes, dark connective tissue, basically. So
And
this is a path,
arthritis.
from tyrosine, it can be converted to fumarate. Fumarate can go to your TCA cycle. All right. So alkaptonuria, as I've said, dark pigment, ochronosis. That's the word that I'm looking for. Ochronosis. The pigment is deposited in connective tissue. So what's the finding? Dark urine when left standing. How are you going to contrast alkaptonuria with porphyria? What's the difference?
Porphyria is red.
Porphyria, like for example, acute intermittent porphyria or porphyria cutanea tarda, right? Anyone?
So you're saying if the test shows dark urine after standing, how do we differentiate?
Yes.
Can we test for heme in the blood?
Okay. You can do test for heme. But the point here, I think, is men and women are equally affected in alkaptonuria, the distribution, right? This is an autosomal recessive disorder. Correct. Compare that to the porphyrias, which is predominantly male, X-link. Okay? That's where I'm going. I'm going into the genetics question, which is 5% of your step 1. Okay, so alkaptonuria, classic finding, dark urine when left standing. What can we see? Arthritis, severe arthritis. You're gonna have black pigment in your cartilage and your joint. Okay, so dark urine when left standing. Again, we talk about MAO and we talk about COMT. Talked about the end product, vanillyl mandelic acid, which is increased in what disorder?
Pheochromocytoma.
Excellent, pheochromocytoma. Where in the adrenal gland is it located? Cortex or medulla?
Medulla.
Medulla. Excellent. What can you give in someone who needs surgery? What can you give? What medicine can you give?
Phenoxybenzene.
Phenoxybenzene. Reversible or irreversible?
Irreversible.
Irreversible. Your phenoxybenzamine. Alpha 1 antagonist, alpha 2 antagonist. alpha 2, right? Antagonist. Symptomatic treatment of pheochromocytoma after surgery, what can you give?
Phentolamine.
Phentolamine, which is a reversible inhibitor of your COMT and MAO. This is your alpha 2 antagonist. Also, don't forget hypertensive crisis. If someone has been taken MAO inhibitors, for example, phenelzine, and they went out, they drank some wine, they eat cheese, they have hypertension, they're having hypertensive crisis, you can give phentolamine. Voila, increase in metanephrine for pheochromocytoma. We have increase in metanephrine and normetanephrine. Check for vanillomandylic acid in the urine. High yield, put a star here. Amino acid derivative of each. Don't forget that tryptophan can be converted into niacin. This is used for the synthesis of niacin, serotonin, and melatonin. Niacin, give me a deficiency in niacin. A disorder of niacin. What is that? Pellagra. Pellagra, the 3 Ds of pellagra, what are those?
Dermatitis.
Go ahead.
Diarrhea.
Go ahead. And the last one?
Dementia.
Dementia. And the last one is death. But let's not talk about that one. So that's niacin. Serotonin. Serotonin is very important. You're going to learn in the next module about your Raphe nuclei, which produces your serotonin. This is very important in pathology of your depression, right? Serotonin. Melatonin is important in what? Sleep, right? So you can give a melatonin agonist like Rameltion to help them sleep better, or Zolpidem, which increases GABA. Increases GABA in your CNS, which increases chloride into the cell, thereby decreasing the transversion or the travel of the action potential, which makes you sleepy, actually. So histidine can become histamine, glycine can become heme, arginine can become creatinine, urea, nitric oxide, glutamate, GABA. GABA is the major inhibitory neurotransmitter in the CNS. Branched chain amino acid, maple syrup urine, homocysteine, homocystinuria, and cysteine, cystinuria. Put a star here. This is high yield amino acid precursor of your neurotransmitters and other good stuff. So for carcinoid syndrome, this is caused by GR tumor. Don't forget you have altered tryptophan metabolism. Tryptophan is a precursor for your niacin. So you can have tryptophan deficiency, pellagra. You can also have serotonin effects because it is made up of tryptophan, your serotonin. So you can have diarrhea, you can have FGF, fibrogenesis, and then you can have flushing. That's carcinoid syndrome. How are we going to break down serotonin? Via monoamine oxidase. So MAO inhibitors, most commonly used in depression. So in the urine, you can see increased high yield in urinary 5-HI, AA, or hydroxyindole acetic acid in carcinoid syndrome. Heart valve disease, again, think about tryptophan, think about niacin. There is an absence of a transporter in your proximal tubule. So you cannot reabsorb tryptophan. So tryptophan is lost in your urine. This is a symptom from your niacin deficiency. Look at the picture on the right. This is pellagra. You can have hyperpigmented rash, red tongue, glossitis, diarrhea, and vomiting. The 3 Ds, dermatitis, diarrhea, and dementia. How are we going to treat? Give them protein, high protein diet, or supplement with niacin. Maple syrup urine disease, we talk about this one, alpha keto acid dehydrogenase. You have an increase in branched chain amino acid and alpha keto acids in your plasma. This is autosomal recessive. So what's the restriction? Restriction of branched chain amino acid. You can't give isoleucine, leucine, and valine. You monitor them. You monitor the plasma amino acid concentrations. And of course, you can give thiamine supplementation. Okay, another high yield point here. Put a star on this slide. Homocysteine. So homocysteine can be converted to methionine via methionine synthase. So you need B12 for that one. Vitamin B12, or homocysteine can be converted to cystathionine. Via cystathionine synthase B6, you need pyridoxine as a cofactor to be converted to cysteine. All right, so the point here is this. Homocysteine can be converted back to methionine. You need B12 for that one. Or it can be converted to cystathionine to eventually be converted to cysteine. So you have cystathionine synthase via B6. Why is this important? Because there's a disease called homocystinuria. Homocystinuria, you have a lens dislocation. Is it, where is the lens dislocated? Superotemporal or inferomedial? In terms of a dislocation of the lens, in Marfan, you also have dislocation of the lens. How are we going to differentiate the lens dislocation in Marfans from homocystinuria?
I don't think.
I think in homocystinuria. Sorry, go ahead.
Sorry, I don't know.
I think in Marfan it goes up and out, and in homocystinuria it goes down and in.
Excellent. So in homocystinuria is inferomedial, okay? Down and in. Contrast this one with Marfan, it's superotemporal, up and out. You can also see long limbs and chest deformities in homocystinuria, osteoporosis, all right, intellectual disability, blood clots, and early atherosclerosis. What's the most common cause of death in homocystinuria? Stroke and MI. That's the most common cause of death due to those blood clots. Cystinuria. Cystinuria is you have decreased reabsorption of cysteine by your proximal tubule of the nephron. So you have kidney stones, right? So how am I going to prevent cystinuria? Methionine-free diet. You don't give methionine. So in the urinalysis, you can see hexagonal urine crystals for your cystinuria. More of this in term 4. Now, amino acid breakdown. We're good so far? I know it's been a lot. How many hours now? We're good so far?
I'm good.
Yes, I'm good.
All right.
It's a marathon, not a sprint.
Yep.
Oh,
As
yeah.
long as you're good. Okay, let's continue then. So, how are we going to break down amino acid? First, you need to remove the nitrogen. Why? So that we can recycle it, right? So via what enzyme? Transamination. So we have here amino acid alpha ketoglutarate. The amino acid group is passed to glutamate. So glutamate gets the nitrogen. So look here, the amino acid and the alpha ketoglutarate. You are removing, right? You are removing now the NH3. Where does it go? It goes to glutamate here. You're forming a bond here, NH3 bond. You are forming a ketone here in your alpha keto acid, the CO. So NH3 is gone. You are now forming a keto, alpha keto acid. Where does the NH2 go? It goes to glutamate. Then what happens? The glutamate can either transfer the nitrogen 2 ways, right? Glutamine synthesis or alanine cycle. That's important part. Glutamate can transfer the nitrogen to the liver via 2 pathways, glutamine synthesis and alanine cycle for excretion in the urea cycle. Let's talk about the glutamine synthesis. So glutamate has the NH2. It transfers the nitrogen to the liver for excretion. So it can either form glutamine, right? It transfers the NH2 to glutamine, and then the glutamine will gonna go to the liver for excretion in the urea cycle. Okay? What's the enzyme here? Glutamine synthase. Another way, right? So in the liver, glutamine is converted back to glutamate. So glutamine here, it's now in the liver. It can be converted back to glutamate. You are releasing the NH3, you are giving it back to glutamate, and then the glutamate via glutamate dehydrogenase will gonna remove the NH2, right? And then this one, the NH, the nitrogen part, will gonna go to the urea cycle, and the remaining part of the glutamate, it just becomes alpha ketoglutarate. Okay, that's the glutamine synthesis. We're good so far. So we are just trying to find ways in which the nitrogen can go to the urea cycle. So it can either be via glutamine synthesis or alanine. So in the glutamate has the NH3, right? It now has the NH3. It needs glutamine synthetase to form glutamine. The glutamine will gonna go to the liver. So you're transferring the NH, the ammonia from glutamate to glutamine. The glutamine goes to the liver. From the liver, the glutamine donates the NH3 to glutamate. It is converted back to glutamate via the enzyme glutaminase. And then the glutamate is cleaved by glutamate dehydrogenase. It removes the NH, the ammonia, to go to the urea cycle. What's left is alpha ketoglutarate. Okay, that's a glutamine synthesis. How about alanine? In the muscle, what happens? So you have glutamate and pyruvate. So it transfers a nitrogen to the liver. So look at the nitrogen here in the glutamate, right? Where does it go? Via your ALT, it goes to alanine. The alanine is now carrying the ammonia, the NH2. Okay, so what happens next? So from alanine to the liver to make pyruvate, and then the nitrogen is transferred back to glutamate. So the NH2 is transferred back to glutamate here. And then from glutamate, the nitrogen is removed from glutamate via glutamate dehydrogenase. The NH3 plus will gonna go to the urea cycle, and the remaining of the glutamate becomes alpha ketoglutarate. So question, between the two syntheses, your alanine cycle and your glutamine synthesis, what is common? What is the common in the two of them?
The alpha ketoglutarate.
The production of alpha ketoglutarate and what else? Glutamate, right? You have the glutamate. The nitrogen is always removed from the glutamate via glutamate dehydrogenase, right? If you look here, glutamate and glutamine synthesis, right? That's the end. That's the most common in there. Glutamate is the last one, the last that gives off the NH2 via glutamate dehydrogenase. Look at the alanine cycle. The last part is always the glutamate. You need to remove the NH2 from the glutamate via glutamate dehydrogenase. What is different from them is the intermediate. In the alanine, you have the nitrogen from the glutamate is given to alanine. When it goes to the liver, the alanine will give it back to glutamate. Contrast this one with your glutamine synthesis, where glutamate will gonna give the NH3 to glutamine. The glutamine goes to the liver, and glutamine is converted back to glutamate. So the only difference is the intermediate. In the glutamine synthesis, you have glutamine. In the alanine cycle, you have the alanine. That's a difference. What's common is the last part. They all end in glutamate donating the NH2, right? They all end up with glutamate dehydrogenase cleaving that NH2 so that the NH2 or NH3 plus in this case, because you're removing the bond, right, can go to the urea cycle. And in the end process, the glutamate becomes, when you remove that NH2, it no longer has that, becomes an alpha ketoglutarate. Okay, those are the high yield points. So now we have the NH3 plus. Where does it go? Urea cycle. High yield, first reaction, and second in the mitochondria. The rest is in the cytoplasm. What's the rate limiting step? CPS1, carbamoyl phosphate synthetase 1. Remember, CPS2 is in your pyrimidine synthesis. High yield N-acetylglutamate is an activator of your CPS1. The enzyme will not function without this cofactor. Okay, so this is synthesized from your glutamate and acetyl-CoA. If you are, if you're in the fed state and you increase your protein, you are activating this N-acetylglutamate, right? This is used to regulate your urea cycle. Oops. So what happens now? So you have NH3 plus here, carbamoyl phosphate synthase 1, carbamoyl phosphate, and then from carbamoyl phosphate to citrulline via OTC. Okay, then citrulline, it will gonna get out of the mitochondria to the cytosol for the rest of the urea cycle. There it is, carbamoyl phosphate, citrulline, arginosuccinate, arginine, urea, and ornithine. But the questions are going to be clinical, right? How are we going to differentiate OTC deficiency from the rest? If you have hyperammonemia, think about urea cycle disorders, okay? If in the vignette you can see flapping of the arms, that's asterixis. Think about urea cycle disorders. What can you give in someone who has hyperammonemia? What's the drug that you can give? Lactulose. Okay, you give lactulose to reduce ammonia reabsorption in the gut. More of that in term 4. Okay, urea cycle. Let's go to vitamins. So B1, this is a cofactor for your dehydrogenases and trans-ketolase. B2 is riboflavin, right? Not that important, but this is for your FADH2, for the electron transport chain. Niacin is important. Don't forget pellagra, right? And heart nut disease for your niacin. Pantothenic acid is important for CoA, for coenzyme A. B6, pyridoxine, your pyridoxal phosphate. This is cofactor in many different reactions. Think about decarboxylases. They need B6. They also use in amino transferase reactions for amino acid, or AST. B7, biotin, for carboxylation enzymes. Anything that has carboxylase there, think about B7. Think also about avidin in raw egg whites, which inhibits the absorption of biotin. There it is, dermatitis, glossitis, loss of appetite, nausea. Folate and vitamin B12. Deficiency, used in synthesis of thymidine, right, for your DTLMP. Both used in metabolism of your homocysteine. So if you have a deficiency on both, you can have decrease in DNA synthesis and increase in homocysteine. So homocystinuria is increase in both. You can have homocystinuria in both deficiency. So what's the clincher? The clincher is methylmalonic acid. Only in vitamin B12, you can have an increase in methylmalonic acid. Learn the pathway of the purine and pyrimidine synthesis, because when you go to term 4, you're going to learn about the drugs, right, that inhibits the enzyme. Vitamin A, we're almost there, we're almost done. Retinoids, retinol, this is important for vision. When you think about vitamin A, think about vision. So you can have night blindness and xerophthalmia. You can have keratinization, you have the thickened dry skin and growth failure in children. Vitamin C are ascorbic acid. This is water soluble. It has an antioxidant properties. Collagen synthesis. What is the function of vitamin C in collagen synthesis?
Hydroxylation of proline and lysine.
Excellent. Hydroxylation of proline and lysine residues for your vitamin C. Also absorption of iron. Don't forget, if you have an increase in vitamin C, you can absorb more iron. So you can develop hemochromatosis. Careful there. Vitamin D, From sunlight, right? So let's do vitamin D synthesis. Who can tell me? Vitamin D synthesis from term 1. So 7-dehydrocholesterol, I'm gonna start you with that, and then cholecalciferol through 25-hydroxy D3. What is that?
In the liver, 25 hydrolase.
So you have your 25-hydroxylase, right? And then in the
One
kidney,
hydroxylase from the kidney.
yes, calcidiol to calcitriol via your 1-alpha-hydroxylase. PTH acts on that enzyme. So when you're doing this now, I recommend you to integrate materials from term 1. Okay, it will be very helpful for you when you go to term 4 and term 5 when you're learning pathology. So learn to integrate as much as possible. But for the lack of time, I'm gonna go fast. So vitamin E, it's an antioxidant. It protects our BC from oxidative damage. Vitamin K, warfarin inhibits vitamin K. What is the function of vitamin K in production of your clotting factors?
It assists the maturation of the cofactors.
Okay, so
The clotting.
the clotting factors, right? So what is that? It carboxylates your gamma residues. Gamma carboxylation of clotting factor residues, I think, if I remember correctly. So don't forget your clotting factors, 2, 7, 9, 10, C, and S. So post-translational modification. All right. Zinc, very important in wound healing, also in taste, also in sexual development. We're almost there. Fabry's disease, just memorize the classic case, okay? Know the deficiency, know what is accumulating, and know the clinical presentation. Fabry disease, child with pain in hands, lack of sweat, and skin findings. Gaucher disease, deficiency of glucocerebrosidase, accumulation of glucocerebroside. Ashkenazi Jewish descent due to founder effects. Splenomegaly, you have anemia, you have bruising and joint pain and fractures. Niemann-Pick, you have sphingomyelinase deficiency. So here you have cherry red spot together with Tay-Sachs, right? So what's the difference between Tay-Sachs and Niemann-Picks? In Niemann-Picks, you have hepatomegaly. In Tay-Sachs, you don't have hepatomegaly. That's the difference. They have the same cherry red spot. In Niemann-Picks, you have hepatomegaly. In Tay-Sachs, no hepatomegaly. How about Krabbe's? You can have progressive weakness, absent reflexes, and optic atrophy. Tay-Sachs, as I've said, most common in Ashkenazi Jewish. You have a cherry red spot, however, no hepatomegaly. Hurler, alpha L hydroronidase, you have accumulation of heparan and dermatan. Surface, so you can have mental retardation, short stature, hepatosplenomegaly, you have coarse features. Contrast that one with Hunter, deficiency of iduronate 2 sulfatase. No corneal clouding in Hunters. Hunters see clearly. It's X-linked. Hunter marks the X. You have behavioral problems. You have learning difficulty. You can have aggressive behavior. That's Hunter's syndrome. And we talk about the different glycogen storage diseases. Any questions?
No.
Oh, by the way, about porphyria, the last one. What's the difference between acute intermittent porphyria and porphyria cutanea tarda? How are we going to differentiate them aside from the enzymes that's deficient? Which one is porphobilinogen deaminase? AIP, right? bilinogen is the cutaneous tarda. So if there is a cutaneous defect, right? If the skin is affected, think about cutaneous tarda. If it's just like mild symptoms, think about AIP, like nausea, vomiting, like that, simple symptoms, AIP. If the skin is involved, think cutaneous tarda, okay, PCT. All right, thank you very much, everyone. Good luck on your exam. Stay safe and healthy. If you have questions, you can DM me. You can comment on the Facebook page. Thank you very much. Have a good day.
Thank you.
So
teacher.
much.
Thank you.